Somewhere in a laboratory freezer sealed in a tiny plastic vial, there's a fragment of bone so small you could lose it in the folds of your palm. No face, no name, no grave marker, and yet locked inside that fragment is a message from a human being who lived tens of thousands of years ago. A message that says, quite clearly, you don't know me. You never knew I existed, and until very recently, neither did anyone else on Earth. That's the strange, almost unsettling truth about human evolution right now. We used to think we understood our family tree. Modern humans on one branch, Neanderthals on another, maybe a few extinct cousins scattered around the edges. Simple, tidy, wrong. Because DNA, the actual genetic code preserved in ancient bones, teeth, and even dirt, has been quietly rewriting the story, and it keeps finding relatives we never knew we had. Not myths, not legends. Real populations of humans who lived, loved, struggled, and eventually vanished, leaving behind almost no fossils at all. The only reason we know they existed is because their DNA is still here, hiding inside us, inside people alive today, passed down silently for thousands of generations. So here's the question we're going to sit with tonight. Who were they? This forgotten lineage that left no bones, no tools we can confidently point to, no burial sites we've found, and yet left an unmistakable genetic fingerprint. How do scientists even detect a human population that essentially disappeared from the physical record? What clues are hiding in our own chromosomes right now as you're listening to this? And maybe more unsettling, how many other lineages like this are still out there, completely undiscovered? I want to be upfront with you. This isn't a story with one neat answer. This is a story built from genuine evidence, real published research, real geneticists staring at real data. But it's also a story full of open questions, competing theories, and educated guesses that shift every year as new tools get sharper. So as we go, I'll be honest about what we actually know for certain, and what's still just our best current interpretation, because that honesty is part of what makes this so fascinating. We're watching a mystery get solved in real time. Before we go further, if you're listening late at night, or maybe just resting with your eyes closed, let me know in the comments where you're tuning in from, and what time it is right now where you are. I love seeing it, Tokyo just past midnight, Toronto during a lunch break, somewhere in the UK right before bed. It's one of my favourite parts of doing this, and if you'd rather listen without ads interrupting or music breaking the mood, I'm also on Spotify now, completely ad-free and music-free, just voice, just the story. You'll find the link in the description. A 5-star review there is honestly the best way to support what I do, and it helps more people find these stories while they're falling asleep just like you. Alright, let's get into it. Because to understand who this forgotten lineage was, we first need to understand something a little unexpected. How exactly do scientists pull a genome out of a bone that's been buried for tens of thousands of years? And just as importantly, what can that genome actually tell us? And what does it stay frustratingly silent about? Let's start with something that sounds almost impossible. DNA is fragile. It's a molecule, and molecules break down over time. Water, oxygen, sunlight, bacteria, all of it chips away at the long chains of genetic code, until, eventually, there's nothing left but chemical dust. Under normal conditions, DNA doesn't survive more than a few thousand years before it's degraded beyond any hope of reading. So when scientists talk about pulling genetic information out of a bone that's 50,000, 70,000, sometimes over 100,000 years old, they're really talking about finding the rare exceptions, the specific unusual circumstances where decay was slowed down just enough for a whisper of the original code to survive. Cold helps enormously. Chemical reactions, including the ones that destroy DNA, slow down dramatically in low temperatures. That's why so many of our best ancient genomes come from Siberia, from high-altitude caves, from permafrost regions where the ground stays frozen or near freezing for most of the year. Dry environments help too, because water accelerates the breakdown of DNA strands, and stable conditions matter just as much as cold or dry ones. A cave that maintains a consistent temperature for tens of thousands of years, without flooding, without wild swings between freezing and thawing, is basically a natural freezer. It's not glamorous, but it's exactly the kind of unglamorous stability that lets genetic information survive long enough for us to find it. But even in the best conditions, not all parts of the body preserve equally well. If you were designing a time capsule for your own DNA, you probably wouldn't choose soft tissue. It rots too fast. You'd want something dense, something protected, something built by the body to last. And it turns out evolution already built exactly that. It's called the petrous bone, a small, incredibly dense structure tucked inside the skull, part of what houses the inner ear. It's one of the hardest, most compact bones in the entire human body, and because of that density, it does something remarkable. It seals DNA inside like a vault, protecting it from the bacteria and environmental damage that destroy genetic material almost everywhere else. Teeth work similarly. The enamel is tough, mineralized, resistant to decay, and it protects the DNA-rich pulp inside. So when researchers go looking for ancient genomes, these are often the first places they look. Not grand, dramatic bones like a femur or a skull. Just a tiny, dense structure most people have never heard of, quietly holding on to genetic secrets for tens of thousands of years. Now here's where things get delicate. Once you have a bone fragment, you're not just dealing with ancient human DNA. You're dealing with a contamination nightmare. Every person who has touched that bone since it was excavated, every archaeologist, every lab technician, every curious visitor who picked it up decades ago before anyone understood the risks, may have left behind their own modern DNA. And modern DNA is intact, abundant, and incredibly good at overwhelming the faint, degraded ancient signal researchers are actually trying to find. This is why serious ancient DNA labs operate almost like something out of a science fiction film. Full-body suits, face masks, dedicated air filtration systems, surfaces cleaned with bleach, rooms that no unauthorized person is ever allowed to enter. Some labs require researchers to shower and change before working. Every reagent, every glove, every tool has to be essentially sterile of human DNA before it ever touches the sample. Because if contamination gets in, the results aren't just slightly off. They can be completely misleadingly wrong. So let's say the sample survives extraction cleanly. What happens next? The DNA that's recovered isn't one long, readable strand. It's shattered into millions of tiny fragments, sometimes only 20 or 30 base pairs long. Modern sequencing technology reads each of these tiny fragments individually, and then using powerful computational tools, scientists align them against a reference genome, essentially a modern human genetic map, to figure out where each fragment belongs. It's a little like being handed a shredded book, one page torn into thousands of confetti-sized pieces, and trying to reconstruct the story using a copy of a similar book as your guide. Painstaking, slow, but increasingly remarkably accurate. How do researchers know they're not just looking at contamination or lab error? This is where authentication comes in, and it's honestly one of the more elegant parts of the process. Ancient DNA has a specific chemical fingerprint. Over thousands of years, certain types of damage accumulate in predictable patterns, small chemical changes at the ends of DNA strands that essentially act like an age signature. Ancient fragments also tend to be shorter, more broken down than the DNA you'd get from a living person or a recently deceased one. When a sample shows this expected damage pattern and this expected fragment length, alongside clean laboratory controls that show no contamination in the blank test samples, scientists gain real confidence that what they're looking at is genuinely ancient. But even with all of this care, there are hard limits. Ancient DNA has never reliably been recovered from anything older than about 1 million years, and realistically, most usable samples come from the last few 100,000. Hot, humid environments like most of Africa, Southeast Asia and South America destroy DNA so efficiently that entire regions remain almost invisible in the genetic record. Not because people weren't there, but because the climate erased the evidence. And when scientists say no DNA was found, that doesn't mean nothing existed. It often just means the conditions were wrong for preservation. Silence in the data isn't proof of absence. It's simply a gap, and gaps are exactly where our forgotten lineage begins to hide. So before we go hunting for a forgotten human lineage, we need to slow down and ask a deceptively simple question. What does lineage even mean? In everyday language, it sounds straightforward. A lineage is a family line, a branch of the family tree. In textbooks, it feels just as clean. Neanderthals, Denisovans, modern humans, each one its own branch with a neat Latin name. But when you look at the actual genetics, it stops being neat very quickly. Geneticists use the word lineage in a very particular way. It doesn't always mean species. It doesn't always mean a separate kind of human. Most of the time, it simply means a group of DNA sequences that share a common history, that split from other sequences at some point in the past and have been evolving along their own path ever since. You can have a lineage of mitochondrial DNA, passed down mostly through mothers. You can have a lineage defined by a stretch of chromosome that looks unusually old. You can have a lineage of Y chromosomes that all trace back to one man tens of thousands of years ago. None of those automatically map onto neatly labelled fossil species. They're just threads in the bigger tapestry. Species, on the other hand, is a biological label, and even that is messier than most of us were taught. Traditionally, a species is a group that can't or doesn't normally successfully interbreed with other groups, but Neanderthals and modern humans interbred. Denisovans and modern humans interbred. There are hints that even more ancient humans mixed in as well. So were they different species, subspecies, regional populations. The biology doesn't care what we call them. Our names are just attempts to draw lines across something that behaves more like a gradient. Then there's population, a word genetics uses in yet another way. A population is a group of individuals that tend to mate more with each other than with outsiders. That can be a band of a few dozen hunter-gatherers or a continental-scale network stretching across thousands of miles. Populations can belong to the same species. They can share many of the same lineages of DNA. They can merge, split, and merge again. In the genetic record, they blur into each other like colours in a watercolour painting. This is why human evolution, when you look at it purely through DNA, doesn't really look like a simple branching tree. It looks more like a braided river. Imagine streams separating, wandering off in different directions, then joining again, then splitting once more. Populations drift apart, accumulate differences, then come back into contact and exchange genes, over and over, for hundreds of thousands of years. This braided pattern is driven by two things: gene flow and chance. Gene flow is just the technical term for genes moving between populations. If a group of Neanderthals and a group of early modern humans meet in the same valley, and some of them have children together, that's gene flow. The child's DNA carries bits of both lineages, braided together. If that child's descendants spread, the genes spread too. One encounter, a few families, and suddenly a whole region's genomes carry faint echoes of a meeting nobody remembers. Chance in genetics has its own word, drift. Genetic drift is what happens when random events shape which genes survive. A storm wipes out a camp. A disease hits one family harder than another. A small group decides to move to a new valley. It's not selection, it's not better genes winning. It's just statistics in small numbers. This is where something called effective population size comes in. It sounds abstract, but it's actually very intuitive. The effective population size isn't the head count of living humans at a given moment. It's the number of individuals who are actually passing genes on in a way that shapes the long-term gene pool. You can have a continent with tens of thousands of people, but if they're scattered in tiny, semi-isolated bands, the effective population size can be much smaller. That makes drift powerful. A new mutation that appears in one person in a small isolated group can, just by luck, spread through that group and then, through migration, into many others. A handful of successful lineages can end up dominating the genetic record, even if they began in groups that would barely register in a global census. Bottlenecks are an extreme version of this, when a population crashes to a tiny size and then recovers. A few surviving lineages get stamped into all future generations. Founder effects are similar. A small group leaves a larger population, carries only a subset of its diversity, and becomes the ancestors of everyone in a new region. Over time, these events carve deep signatures into DNA. They can make a minor side stream in the braided river look enormous, simply because it ended up downstream of everything else. This has an important consequence for our forgotten lineage. When geneticists say they see evidence of a mysterious ancestral lineage in modern DNA, they aren't necessarily saying there was a whole separate species of humans with their own name and their own skull shape. They're saying that some chunk of our genome seems to have come from a population that split off from our other ancestors at a certain time, followed its own path for a while, and then flowed back in. Forgotten can mean different things. It can mean truly extinct, a group that died out entirely, leaving only the genetic traces hiding in our cells. It can mean absorbed, a population that merged so completely with others that, culturally and physically, its distinctiveness vanished, even while its DNA persisted. Or it can mean simply unsampled, a group that left no DNA we can recover, and no fossils we've confidently found, even though they were there, living, breathing, and having children. Here we run into the difference between two very different kinds of gaps. A fossil gap is an absence in the bones and tools we've dug up so far. Maybe the sediments weren't right for preservation. Maybe the right cave is still under a hillside nobody has excavated. Maybe the bones eroded away 10,000 years ago. The Earth is huge, our excavation sites are tiny. The fossil record will always be patchy. A genetic gap is something else. It's a pattern in the DNA we do have that doesn't quite fit the existing branches of the family tree. A stretch of genome that looks too old, or that clusters with neither Neanderthals nor known modern human populations. A signal that says quietly but persistently, there was someone else here. When those two kinds of gaps line up, no fossils, but a strange genetic signal, that's where the idea of a forgotten lineage begins. Not with a name, not with a skull on a museum shelf, but with statistics, probability, and a braided river of ancestry that refuses to be neatly mapped. To see how confusing this can get, we need to step back into a time before we had any ancient DNA at all, when scientists were trying to piece together the human story from bones alone, and already seeing shapes that didn't quite fit the labels they'd been given. For most of the 20th century, the story of human origins was told with two big, competing pictures. One was the classic out-of-Africa model. In its early, simple form, it said that modern humans evolved fairly recently in Africa, maybe 200,000 or 300,000 years ago, and then spread out across the world, replacing all the older humans they met with little or no interbreeding. In this view, Neanderthals, Homo erectus, all the older forms were evolutionary dead ends. Impressive, maybe, but gone. We were the new model, and we took over. The other big picture was the multi-regional model. It said something very different, according to this idea. After an early ancestor like Homo erectus spread out from Africa more than 1 million years ago, evolution continued in many regions at once. Europe, Africa, and Asia all had their own local populations, slowly changing over time, but tied together by occasional gene flow. Modern humans, in this view, didn't arise in one place and spread. They emerged gradually, everywhere, from a network of ancient populations. Neither side had DNA. They had skulls, teeth, fragments of jaws, stone tools, layers of sediment with dates that were often uncertain by tens of thousands of years. And they were trying to build sweeping narratives out of a jigsaw puzzle where most of the pieces were missing. Even then, the bones themselves were sending mixed messages. Some fossils looked comfortably archaic or modern, a thick-browed skull from Europe with a long, low brain case. Easy to call it Neanderthal, a rounder, more vertical forehead from Africa. That felt like an early modern human. But then there were the troublemakers, the ones that refused to fit the neat categories. Scientists started finding skulls and skeletons with what they called mosaic traits, a face that looked relatively modern, combined with a very thick, archaic brain case. A small, light skeleton, but with massive brow ridges. Teeth that looked transitional, halfway between what we expected from early Homo and from later forms. In East Africa, in places like Omo and Herto, you get skulls that look almost like us, but not quite. In North Africa, at Jebel Irhoud, fossils first thought to be Neanderthal-like were later redated and reinterpreted as some of the oldest candidates for early Homo sapiens, yet with features that seem surprisingly primitive. In China, fossils like those from Dali or Jinyu Shan showed a mix of archaic and more modern traits that didn't match European Neanderthals or African early moderns very well at all. Depending on which model you favoured, you could read these mosaics very differently. If you leaned out of Africa, you might say, these are early or transitional modern humans, still shedding archaic features as they spread. If you leaned multi-regional, you might say, these are regional forms, shaped by long local evolution and occasional mixing with neighbours. Underneath the labels, though, there was a deeper unease, because some of these fossils didn't just look in between. They looked like they might represent side branches, populations that had split off, gone their own way for a while, and maybe interacted again later. But without DNA, that was hard to prove. You had shapes, not lineages. Then there was the problem of time. Every fossil sits in a layer of sediment, and that layer has to be dated. Early methods were rough. Some relied on the kinds of animals buried nearby, or on the types of stone tools found in the same layer. Later, radiometric techniques like radiocarbon, uranium series, argon-argon, let scientists put actual numbers on ages. But each method has its limits. Radiocarbon fades out beyond about 50,000 years. Uranium dating can be thrown off by groundwater. Argon methods need volcanic layers in just the right place. So a skull that was once thought to be, say, 100,000 years old might later be revised to 200,000, or down to 60. As dating techniques improved, timelines shifted. Fossils that seemed too early to be modern humans turned out to be younger, suddenly fitting more comfortably into an out-of-Africa frame. Others went the opposite way. Bones, once considered relatively recent, were pushed back in time, making them awkward for any simple recent replacement story. Geography made things even harder. Fossils preserve best in cool, dry, stable environments. Caves, lakeside sediments, places with gentle burial, and little chemical destruction. Huge parts of the world don't offer those conditions. Tropical forests, with their heat, moisture, and acidic soils, are terrible for long-term bone survival. Desert sands can erode and scatter remains, or bury them so deep that nobody ever finds them. Mountain slopes wash fossils downslope or grind them away. That means that vast regions of Africa, South and Southeast Asia, much of Island Asia, large chunks of Arabia, places that were almost certainly full of ancient humans, are nearly blank in the fossil record. Not because nobody was there, but because bones simply didn't last or haven't yet been found. Yet archaeology, the record of what people did, sometimes suggests meetings we can't see in the bones. Stone tool traditions appear in sequences. One type of flaking style gives way to another. Suddenly, in a region that had been using simple core and flake tools for hundreds of thousands of years, a more sophisticated, standardized technology appears that looks suspiciously like what's been found 1,000 miles away. Sometimes it shows up abruptly without a clear local transition. You can explain that by independent invention. But often the pattern looks more like cultural contact, people moving, or at least ideas moving, across long distances. A new style of spear point, a new way to prepare stone cores, a new approach to hafting blades to handles, all spreading faster than slow, skeletal evolution would predict. This created a strange tension. The bones weren't telling a complete story. The tools hinted at connected networks, the dates kept shifting, and certain fossils, especially those with very archaic features in unexpectedly recent layers, or very modern features surprisingly early, seemed to whisper that there had been unsampled populations in the mix. By the late 20th century, some anthropologists were already talking about unknown ancestors and ghost populations, even if they didn't use those exact terms. They would say things like, "This skull looks like it has input from a population we haven't yet found." Or, "This Asian fossil doesn't match known Neanderthals or African moderns. Maybe there was a local lineage, now lost." Others pointed to odd combinations of traits in European and Near Eastern fossils, and wondered whether Neanderthals had interbred with some other archaic group before modern humans ever arrived. These were cautious speculations, framed in the language of morphology and regional continuity. But the basic idea was clear. The human story probably involved more players than the named species on the museum labels. Without DNA, those extra players remained shadows. Hypotheses in the margins of papers, phrases like possible admixture, regional variant, unresolved affinity. The fossil puzzle didn't fit neatly. There were pieces that seemed to belong to branches we hadn't drawn yet. There were hints of lineages that must have existed to explain the shapes we saw, even though no one had pinned a definitive name on them. Then a tiny fragment from a Siberian cave changed what was possible. When scientists learned how to pull genomes out of ancient bones, those shadowy, predicted ancestors started to step into the light. Denisova Cave, in the Altai Mountains of Siberia, had been known to archaeologists for years before anyone realized what was hiding there. It's a good cave, from an archaeologist's point of view, high up, dry, with deep layers of sediment laid down over hundreds of thousands of years. People had come and gone many times. They left stone tools, bits of bone, traces of fires. Excavations in the late 20th century had already turned up Neanderthal remains there. So the cave fit comfortably into the familiar picture. One more cold climate shelter on the eastern fringe of the Neanderthal world. Then, in 2008, a tiny fragment came out of the ground. It wasn't impressive. A child's finger bone from the last segment of the little finger. No skull, no jaw, no dramatic brow ridges or distinctive chin. Just a sliver of phalanx that, on morphology alone, couldn't be confidently assigned to anything. It didn't scream new species, it barely whispered interesting, but by that point something new was available, reliable techniques for extracting and sequencing ancient DNA from very old, very degraded bones. The team working on Denisova Cave sent the little bone off for genetic analysis. The expectation was modest. Maybe it would confirm that this was a Neanderthal child. Maybe if they were lucky, the DNA would be good enough to say something about population structure, or about how closely related Eastern Neanderthals were to the better-known Western ones. When the first results came back, they did not fit the script. The mitochondrial DNA, the small ring of genetic material passed down the maternal line, didn't look like that of Neanderthals, or like that of modern humans. It was closer to Neanderthal than to us, but clearly separate. A distinct branch. That alone was surprising. Mitochondrial DNA is only a tiny fraction of the genome, and it can be quirky. But when researchers went on to sequence much more of the nuclear genome, the surprise hardened into something that couldn't be waved away. The child from Denisova Cave belonged to a population that was neither Neanderthal nor modern human. Close cousins, yes. Sister lineages, yes. But genetically distinct in a way that said, this isn't just a regional variant. This is a separate branch of the human family tree. To make sense of that, you have to think about how geneticists estimate divergence times. The basic idea is simple in principle. Mutations accumulate over generations. If you compare the genomes of two populations and count how many differences there are, you can get a rough sense of how long they've been evolving separately, assuming you have some estimate of how fast mutations usually happen. In practice, it's messy. Mutation rates are not perfectly constant. Different parts of the genome evolve at different speeds. Population sizes and structures matter because they affect how quickly new mutations spread. So instead of exact dates, you get ranges, probabilities, windows in time rather than calendar days. When scientists did these calculations for the Denisova child's genome, they came up with something like this. The lineage that led to Denisovans and Neanderthals split from the lineage that led to modern humans roughly 600,000 to 800,000 years ago. Then, later, Denisovans and Neanderthals split from each other, perhaps 300,000 to 400,000 years ago. Those numbers are not fixed in stone. Different studies, using different assumptions and calibration points, shift them up or down by tens of thousands of years. But the overall pattern is stable. Modern humans on one branch, Neanderthals on another, Denisovans on a third, all meeting back in a common ancestor hundreds of thousands of years in the past. The finger bone had revealed a whole lineage that nobody had been able to point to clearly in the fossil record, which raised an awkward question. How had we missed them? Part of the answer is geography and preservation. Much of Asia is bad for long-term fossil survival, and fieldwork coverage is patchy. But there's another issue. Without DNA, bones are identified by shape, and there is a lot of overlap in shape among closely related human populations, especially if you only have fragments. A child's finger bone looks like a child's finger bone. Even a skull can be ambiguous if it has a mix of archaic and more modern features. Some Asian fossils found in the 20th century, the mosaic ones that didn't fit neatly as Neanderthal or early Homo sapiens, might belong to populations related to Denisovans or to other unsampled branches. But on morphology alone it was hard to say. The category Archaic Asian Homo became a kind of catch-all for things that didn't look European Neanderthal or African modern. When the DNA from Denisova Cave finally came out, the name Denisovan was simply a practical label based on the site. It doesn't mean a fully described species in the classical anatomical sense. it doesn't necessarily correspond to a particular skull shape or a specific set of visible traits. It is, first and foremost, a genetic cluster, genomes that group together, distinct from Neanderthals and from us. Over time, a few more pieces joined that cluster. A large molar from the same cave, with surprisingly big, complex cusps, yielded matching DNA. A jawbone from the Tibetan Plateau, too degraded for full genome recovery, preserved enough protein fragments to link it to the Denisovan branch. But compared to Neanderthals or modern humans, the physical remains are still vanishingly sparse. Denisovan, then, is a reminder of how lopsided our knowledge is. A lineage defined more by sequences of A, C, G, and T than by rows of display cases in museums. A population we know intimately in genetic terms, but can barely visualize in skeletal form. And that was the real shock. If a whole branch of our family tree could hide in plain sight, masked by fragmentary fossils and lost in the noise of archaic categories, what else might be out there? How many other lineages had left their mark in DNA, but not yet in named bones? The Denisovan discovery didn't just add a new cousin to our story. It opened the door to a new way of thinking, that ancient genomes might reveal entire populations we have never seen, shadows in the statistics that hint at forgotten humans sharing the world with us. Once you have more than one branch on the family tree, another question appears almost automatically. Did they stay separate? Or, when their ranges overlapped, did they meet in the only way populations really meet in evolutionary terms, by having children together? For a long time that was debated using skulls and stone tools. People argued from the thickness of brow ridges, the slope of foreheads, the style of spear points. Some thought Neanderthals and modern humans were too different to have viable offspring. Others suspected mixture, but had no way to prove it. Ancient DNA changed that. Not just because you can sequence Neanderthal and Denisovan genomes, but because of what you can see when you compare those ancient genomes to people alive today. If you take the DNA of a person with European or Asian ancestry and line it up against a Neanderthal genome, something strange happens. Certain stretches of the modern genome are noticeably more similar to Neanderthal than they are to other modern humans. Not just by chance, not just a few letters, whole segments. These are called introgressed segments, pieces of DNA that came into one population from another through interbreeding in the past. You can think of your genome as a long, braided rope. Most of the strands are inherited from within your own lineage, but if, at some point, your ancestors had children with members of another lineage, a few strands from that other rope get woven in. Over generations, the ropes are spliced and respliced, but those foreign strands can still be traced, if you look closely enough. So how do geneticists actually see those strands? One of their favourite tools has a rather whimsical name, the ABBA-BABA test. In the technical literature, it's wrapped up in something called D-statistics, but the underlying logic is surprisingly simple. Imagine 4 genomes arranged in a little family tree. On one side, a modern human from Africa who is unlikely to have recent mixture with Neanderthals. On another branch, a modern human from, say, Europe or East Asia. On the third branch, a Neanderthal. On the fourth, a more distant outgroup like a chimpanzee, used as a reference for what the ancestral state of a DNA letter probably was. Now focus on a single position in the genome where there are 2 possible versions of a letter. Let's call them A and B. Suppose the chimp has A, the African human has A, but both the European human and the Neanderthal have B. That pattern, A, B, B, A, is called ABBA. If instead the chimp has A, the European human has A, and the African human and Neanderthal share B, that's BABA. If there had never been any mixture between Neanderthals and any modern human population, you'd expect ABA and BABA patterns to occur at roughly equal rates, just due to the random branching of lineages over time. But if the European human has inherited some DNA from Neanderthals, you'll see more ABA than BABA, more sites where the European and Neanderthal line up together against the African. Count up those patterns across millions of positions in the genome, and the imbalance becomes a signal, not a vague impression, but a statistically robust tilt. Enough to say, these two lineages shared genes after they split. The same kind of logic works for Denisovans. Replace the Neanderthal genome in that four-way comparison with a Denisovan genome, and you can ask, do people in, say, Papua New Guinea, share more derived variants, more Bs with Denisovans than people in Africa do? The answer is yes. Strongly yes. That's introgression. Once you accept that mixture happened, another layer of information opens up, because those introgressed segments don't stay intact forever. Every generation during the formation of eggs and sperm, recombination shuffles the genome. Chromosomes from your mother and father line up, swap chunks, and pass on a patchwork to the next generation. Over a handful of generations, a segment of Neanderthal DNA might still be a long, continuous stretch. Over thousands of generations, that stretch gets chopped into smaller and smaller pieces, scattered around the genome like confetti. That means the length of introgressed segments carries a kind of clock. Big, unbroken pieces suggest more recent admixture. Tiny, fragmented bits suggest very ancient events. By measuring how long those Neanderthal-like or Denisovan-like segments are in modern genomes, and how many breaks recombination has had time to make, researchers can estimate when the mixing probably occurred. For Neanderthal ancestry in non-African modern humans, the dates cluster around 50 to 60,000 years ago, consistent with the first major expansions of our species out of Africa. Denisovans signals in different regions of Asia and Oceania point to several separate pulses, not just one romantic evening on a Pleistocene steppe. And that's another key point. When you see introgression, it rarely traces back to a single tidy episode. Different populations can meet and mix multiple times, in different places, under different circumstances. Later waves of migration can carry those blended genomes even further, overlaying one pattern of admixture on top of another. So when geneticists say this population has Denisovan ancestry, they don't usually mean one event. They mean that, when you unravel all the ropes and map all the segments, the best explanation involves several contacts, possibly separated by tens of thousands of years. Of course, not every signal that looks like gene flow actually is gene flow. There's an important alternative that keeps geneticists awake at night: population structure. Imagine a single widespread species, divided into semi-isolated regional groups for a very long time, without ever fully splitting into separate species. Those groups will accumulate their own local mutations, simply because they're more likely to mate within the group than outside it. Hundreds of thousands of years later, descendants of one region might share more variants with a particular fossil genome, not because their ancestors interbred with that fossil's population, but because both trace back to the same structured patchy ancestor. Distinguishing ancient structure from later admixture is hard. It involves modelling many possible histories, checking which patterns of shared variation they would produce, and seeing which model matches the data best. And even then, there is uncertainty, confidence intervals, caveats in the fine print. So when you hear that modern humans have 2 to 4% Neanderthal DNA, or that some Indonesians carry Denisovan ancestry, it's worth remembering how much statistical and conceptual machinery sits behind those numbers, and how much is still being debated. There's another misconception worth setting aside as well. Interbreeding doesn't imply harmony. The fact that genomes mix tells us that, at least some of the time, individuals from different lineages formed bonds close enough to produce children who could survive and reproduce. It doesn't tell us what the overall relationship between those groups was like. Contacts could have ranged from peaceful exchange and shared campsites, to kidnapping, to brief encounters at contested borders. Over tens of thousands of years, all of those probably happened. What the DNA does show is that the boundaries between lineages were porous, not absolute. Neanderthals, Denisovans, and early Homo sapiens were not completely sealed worlds. They were part of a wider network of hominins meeting and parting across ice sheets, forests, and mountain passes, leaving traces of those meetings in the chromosomes of people alive today. And sometimes, when you zoom in on those traces, you see something even stranger, patterns that don't quite fit any known ancient genome, signals of mixture with a population we haven't yet sampled at all. A ghost in the statistics, hinting at a forgotten lineage that once walked alongside Neanderthals, Denisovans, and us. When geneticists talk about a ghost population or a ghost lineage, they don't mean anything supernatural. They mean a group of real flesh and blood humans that once existed, left descendants, but has not yet been directly sampled. No skull in a museum, no jawbone in a drawer, no genome neatly sequenced from a finger bone. Instead, its existence is inferred from patterns in the DNA of people we have sampled. It shows up as a mismatch between what we expect, given the fossils and genomes we know, and what the statistics stubbornly insist is there. To see how that works, imagine that family tree again. You have present-day Africans, Europeans, East Asians, Papuans, a Neanderthal, a Denisovan, and maybe a chimp as an outgroup. If those were the only players in the story, you could in principle find a clean branching pattern that explains who is more closely related to whom and how much mixture happened between each pair. You tune your model. A bit of Neanderthal ancestry in non-Africans here, a couple of Denisovan pulses into island Southeast Asia there. You run your tests, count your ABBA and BABA patterns, and something still doesn't fit. Certain distant populations share unusual variants that shouldn't be there if the only sources of archaic ancestry were Neanderthals and Denisovans. Or an African group seems oddly close, at some positions in the genome, to a non-African archaic sample, even though that population is thought never to have left Africa, let alone met Neanderthals in a European valley. These are the first hints of a ghost. At the level of a single site, it's nothing, just noise, but across millions of sites, small systematic imbalances start to appear. Too many ABBA patterns of one specific kind, clusters of alleles, specific DNA variants that seem to have a deeper history than the rest of the genome. You can think of an allele as a little signature, a way of saying this version of the gene took this particular evolutionary path. If two distant groups share a lot of rare, derived alleles, mutations that arose after they split from other lineages, that's usually a sign of either gene flow between them, or shared ancestry from a particular subgroup in the deeper past. When those shared alleles can't be explained by known fossils, you start to suspect an unsampled branch. But suspicion isn't enough, because missing reference genomes can really distort the family tree. If you only have Neanderthal and Denisovan as your ancient anchors, it's tempting to treat them as representatives of all the archaic diversity that ever existed outside Africa. They're not. They're two twigs on what was once a very bushy tree. Leave out a branch, and your model will try to compensate. It will bend other branches, inflate or deflate mixture estimates, or assign too much importance to population structure inside modern humans to make the math work. You can end up blaming Neanderthals for patterns that actually come from some other now invisible cousin. So how do you tell the difference between a phantom created by a bad model and a genuine ghost population? This is where simulations come in. Researchers build computational models of population history. Who split from whom, when, how big each group was, how often they exchanged migrants, how much they shrank or grew. Then they evolve genomes along those branches on the computer, letting mutations appear, recombinations shuffle, lineages drift. The result is simulated genetic data, born from a specific hypothetical history. You can start with a simple model, modern humans splitting from Neanderthals and Denisovans, some known admixture events, no ghosts. From that, you generate fake genomes and calculate all the same statistics you do on real data. D statistics, allele frequency spectra, patterns of linkage, lengths of introgressed segments. Then you compare. If the simple model reproduces the observed patterns pretty well, you don't need anything more exotic. But often it doesn't. There are stubborn discrepancies, too many deep branching alleles in one group, or unexpected sharing between regions that are supposed to have been isolated. So you add a ghost. You introduce a new branch, maybe splitting off very early from the main human stem, or from the Neanderthal Denisovan line. You let that ghost population persist for hundreds of thousands of years, perhaps mostly in one region. Then you allow it to contribute a small fraction of genes into another population at a particular time in the past. Run the simulation again, generate new fake genomes. Recalculate the statistics. If the fit suddenly improves, if those strange sharing patterns, those extra deep alleles, those odd asymmetries between populations now fall in line, then the ghost is doing real work. It's not just a toy, it's the simplest addition that makes the model match the data. This is what confidence looks like in population genetics. Not a photograph, not a single dramatic bone. Instead, a convergence of many lines of statistical evidence under many different modelling assumptions, all pointing toward the same conclusion. Some unsampled group with certain approximate properties almost has to have existed. Confidence is also bounded. You might be able to say, with some precision, that admixture from a ghost population probably contributed a few % to the ancestors of a particular modern group, and that the split between that ghost and other lineages goes back, say, 700,000 years. But you often can't say where they lived, what they looked like, or what kind of tools they used. And there are always alternative explanations that have to be carefully ruled out. Long-lasting population structure inside Africa, for example, or unrecognized substructure among Neanderthals and Denisovans themselves. It's also crucial to remember that there isn't just room for one ghost. The Pleistocene world was big. Human-like populations were scattered across Africa and Eurasia for more than 1 million years, often in small, semi-isolated groups. There is no reason to think that only 3 or 4 of those groups left descendants. There could have been many lineages that branched off, persisted regionally, mixed here and there, and eventually vanished as recognizable fossils, yet still left fragments of their genomes behind. Different present-day populations might carry traces of different ghosts. One unsampled lineage contributing to the ancestors of West Africans. Another very deep branch feeding into an ancient Denisovan-like group in Asia. A third perhaps mixing with early Homo sapiens in the Middle East before the big expansions out of Africa. From the point of view of the statistics, each of these leaves a slightly different fingerprint, different patterns of allele sharing, different depths of divergence, different segment lengths and frequencies. Piece by piece, those fingerprints are being catalogued. Some ghosts are now widely discussed in the literature, with fairly well-constrained timelines. Others are still hazy suggestions, unexplained deviations in a graph that might fade with better data. Either way, the message is the same. The lineages we can name, Neanderthal, Denisovan, modern human, are only the most visible peaks of a much larger landscape. Between them, beside them, and deep in their roots, Other populations once lived and loved and raised children, long before anyone put a label on them. In some regions, we have their DNA but no bones. In others, we may have their bones but no genomes yet. And in at least one place, the signal of a ghost becomes especially intriguing, because it seems to come from Africa itself, in a time and place where the fossil record is thin and the story of our origins is still being pieced together. If you're going to go looking for ghosts, Africa is the place you have to start. Not because it's mysterious in some vague, romantic way, but because it has the deepest genetic diversity of any continent on Earth. Every comparison of human genomes tells the same story. African populations, on average, carry more variation, more old lineages, more time depth than populations anywhere else. That makes sense. Our lineage has been in Africa the longest. Mutations have been accumulating there for hundreds of thousands of years through bottlenecks and expansions, droughts and green phases. When small groups left Africa 60 or 70,000 years ago, they carried only a slice of that diversity with them. Everyone outside Africa descends from that slice. Inside Africa, much more of the original complexity remained. But that same centrality creates a problem. If we want to read Africa's deep history the way we read Eurasia's, by sequencing ancient bones and comparing them, we immediately run into physics and chemistry. DNA hates heat and humidity. In cold caves and permafrost, genetic material can last for tens of thousands, sometimes even hundreds of thousands of years. In warm, wet, oxygen-rich environments, it breaks down much faster. Enzymes chew it up, microbes recycle it. Water and heat shred the strands into pieces too short and too damaged to make sense of. Most of Africa, for most of the past few 100,000 years, has not been kind to DNA. So, while we now have a growing library of ancient genomes from Europe, Siberia, even parts of high-altitude Asia, the African shelf is still almost empty. There are a few remarkable exceptions, some Holocene skeletons, a handful of late Pleistocene individuals, but they sit very close to the present compared to the time spans we've been talking about. The period when Neanderthals and Denisovans were diverging outside Africa, when the bottleneck survivors were branching inside Africa, when strange archaic cousins might have been living side by side with early Homo sapiens. For that era, African DNA is mostly gone. And yet, the living people of Africa still carry a record of what happened. It's not written in ancient bones, it's written in how their genomes are structured today. When researchers began sequencing whole genomes from diverse African populations, they found something odd. Some groups, particularly in West and Central Africa, seemed to carry stretches of DNA that were unusually old, not just older than the out of Africa split, but older than the split between modern humans and Neanderthals. Certain segments look as if they diverged from other human DNA, 600, 700, even 900,000 years ago, and then somehow persisted, in low frequencies, into the present. Too deep, too concentrated in particular populations, to be easily explained by simple continuous population structure. One way to make sense of this is to invoke the same kind of ghost we met earlier, but this time living inside Africa. In these models, an early branch of the human family tree splits off in Africa long before classic Homo sapiens emerges. That branch hangs on, perhaps in relatively isolated refuges, for hundreds of thousands of years. Then, sometime in the last 50 to 200,000 years, it comes back into contact with the ancestors of some present-day African groups, and contributes a small fraction of their genes. Not 50%, not even 20, more like a few percent, maybe under 10. Just enough to leave a statistical footprint. Different studies propose different timings and depths, and not everyone agrees on the details. Some models say you can explain much of the pattern with long-lasting structure among early Homo sapiens populations themselves, groups splitting and reconnecting inside Africa without needing a very ancient ghost. Others suggest at least one truly deep, archaic branch that never participated in the main Homo sapiens story until late. But in most of these reconstructions, the same general picture keeps reappearing. Africa was not one big, well-mixed population slowly evolving into modern humans. It was a continent full of semi-separate groups, some of them very deeply diverged, occasionally reconnecting and exchanging genes. If that's true, what would we expect to see in the ground? Maybe something like this. You might have fairly modern-looking Homo sapiens skulls in one region, rounded craniums, reduced brow ridges, faces tucked in under the brain case. In another region, at roughly similar times, you might find fossils that look more archaic, thicker brow ridges, longer and lower skulls, more robust jaws. And then in intermediate zones or later layers, you might start seeing skeletons with a mosaic of traits, partly modern, partly archaic. In fact, Africa already has fossils that fit this kind of mosaic description. Skulls from places like Jebel Irhoud in Morocco, Omo Kibish in Ethiopia, Florisbad in South Africa, and several others show combinations of features that don't line up neatly with the classic modern human template. Some look almost modern from the front, but archaic from the side. Others have modern faces but more primitive braincase shapes. A few finds, jawbones, partial skulls, are difficult to classify at all. It's tempting to match these directly to the genetic ghosts, to say this one is the archaic source or that one is the hybrid. But the dating is often uncertain, the fossils are sparse, and without DNA from the bones themselves, those connections remain speculative. What we can say is that the fossil record already tells us that African human diversity 100 to 300,000 years ago was not simple. Different morphologies coexisted. Transitional forms are common. Line drawings of our evolution as a single straight arrow have never really fit the evidence. The genetic hints of archaic admixture strengthen that impression. They suggest that some of those unusual fossils might not just be early modern humans on a straightforward path, but representatives of side branches that later fed back into the mainstream. And yet, the word archaic can be misleading here. When people hear archaic humans, they think of Neanderthals, very distinct, very separate, living in distant caves. But inside Africa, the boundaries between archaic and modern are probably blurrier. Instead of clearly separate species exchanging genes only occasionally, we may be looking at a long continuum of populations, some more isolated, some more connected, all evolving under shifting climates and landscapes. In that scenario, an archaic population in Africa might not have looked dramatically different from its Homo sapiens neighbours. It might have shared many behaviours and technologies. The distinction would be about deep branching times, subtle anatomical trends and genetic histories, not about cartoonish cavemen versus us. This matters for our forgotten lineage because it widens the range of possibilities. The ghost we're chasing might not be a sharply defined exotic cousin like the Denisovans. It could be one of several long-lived African lineages, blending gradually into the emerging Homo sapiens network, leaving only faint, ancient signatures in the genomes of their descendants. To follow that trail further back to the oldest, strangest branches that might have contributed to these ghosts, we have to look even deeper into time, to an era when our ancestors were just one of many human-like creatures on the planet, and some splits may reach almost to the very root of our genus. When geneticists talk about a super-archaic lineage, they're pushing that idea of depth to its limit. Archaic already means something like Neanderthals or Denisovans, branches that split from our own line a few 100,000 years ago. Super-archaic is a way of saying, no, even older than that, a population that diverged from the ancestors of humans Neanderthals and Denisovans so early that, in some models, it's almost brushing up against the boundary between Homo and whatever came before. Not everyone likes the term. It sounds dramatic, like a new species of monster. In reality, it's just shorthand for a very deep split time, a label for a branch that might have separated from our lineage 800,000, maybe over 1,000,000 years ago, and then later mixed back in. Why would anyone think such a thing existed? The pathway to that idea runs through statistics, not skeletons. When you compare genomes, modern humans, Neanderthals, Denisovans, you can estimate how long ago different chunks of DNA last shared a common ancestor. Most of our genome coalesces or comes together within a few 100,000 years. Some stretches go deeper. That's expected. Gene lineages are messy. They don't all merge at exactly the same time. But when researchers started building complex models to fit all the data at once, modern genomes, archaic genomes, population sizes, known splits, they kept bumping into a problem. Certain regions of the genome in some modern populations, and even in Denisovans and Neanderthals themselves, seem to be too divergent, too old, to fit neatly on the family tree without adding something extra. Imagine trying to draw a tree that connects 4 branches, but a few twigs keep landing so far away you'd need a longer trunk. One way to handle that is to say, maybe the trunk is older than we thought. Another way is to say, maybe one of those branches actually came off a different tree and got grafted on later. That second option is the super-archaic hypothesis. In some studies, the best fitting models suggest that Neanderthals and Denisovans themselves may have picked up a small percentage of their DNA from a population that split off long before they did. Perhaps a relic group descended from early Homo erectus in Eurasia. Later, when Neanderthal Denisovan ancestors expanded out of Africa, they met these older residents and interbred. Other work, looking at modern African genomes, hints that something similar might have happened inside Africa, an ancient branch, maybe close to the base of Homo heidelbergensis or another early form, hanging on in isolation, and then contributing genes to the ancestors of Homo sapiens long after the initial split. These are not huge contributions, 1, 2, maybe 3%. But because the divergence is so deep, that small fraction can have an outsized effect on the statistics. This is where the uncertainty starts to balloon. The further back you push your models, the fuzzier the picture becomes. Mutations have had more time to accumulate and get overwritten. Recombination has chopped ancestral segments into smaller and smaller pieces. Ancient population sizes become harder to estimate. Different demographic scenarios, splits, bottlenecks, expansions, can mimic each other's genetic signatures. A pattern that looks like super-archaic introgression under one set of assumptions might look like long-lived structure within a single species under another. Those are the two main competing interpretations. In the introgression scenario, you have a fairly clean tree, a main human Neanderthal Denisovan lineage, plus one or more very old side branches that diverge early and then, much later, contribute a trickle of genes back into the main lines. Think of small streams feeding back into a river after flowing separately for a very long time. In the ancient structure scenario, you don't need separate species. You imagine a widespread ancestral population that was already split into semi-isolated subgroups. Some of those subgroups stayed apart long enough that their local variants drifted a long way from each other. Then, when conditions changed, those subgroups reconnected and mixed. From a genetic point of view, both situations can create very deep coalescence times for some stretches of DNA. Both can produce the illusion of a ghost ancestor. So which is it? Right now, the data don't allow a single confident answer. Different methods emphasize different aspects of the signal. Some favor true super-archaic introgression. Others argue the patterns can be explained without invoking a lineage as old and separate as that. To move beyond this stalemate, we'd need new kinds of evidence. One possibility is older DNA. If researchers could recover genomes from human-like fossils around 1 million years old, early Homo in Africa, or early Homo erectus in Eurasia, we could anchor those deep branches directly. We'd see in the sequences themselves whether Neanderthals or Denisovans carry chunks that clearly match those early forms. That's technically very hard, especially in warm regions, but not impossible in cooler, protected sites. Protein sequences from ancient teeth and bones, the emerging field of paleoproteomics, might provide a partial substitute when DNA is gone. Proteins preserve longer and can still reflect deep evolutionary relationships. Another possibility is a much richer fossil record, tied to more precise dating. If we start finding late-surviving, very archaic human forms in regions where our models predict super-archaic contact, that would lend weight to the idea. Conversely, if the fossils in those regions all look broadly similar to known Neanderthal or early Homo sapiens morphologies, the case for a truly separate, long-lived branch becomes weaker. Improvements in modelling could also shift the balance. As computational methods grow more sophisticated, they might distinguish more clearly between signals caused by introgression and those caused by ancient structure. Right now, the error bars are wide. Why does any of this matter for our forgotten lineage? Because the super-archaic hypothesis forces us to think about the very earliest expansions of Homo, not just the later, better-known ones. If Neanderthals and Denisovans carried genetic traces of a population that left Africa long before them, then the story of out of Africa is not a single migration, or even a handful. It becomes a repeated pattern. Waves of humans and human-like beings moving into new lands, encountering earlier waves, sometimes replacing them, sometimes absorbing them. In that world, our ghost lineage might not just be a late local curiosity, a small side branch feeding into modern Africans. It could be part of a much deeper tapestry, woven from expansions and contacts that started close to the very origin of our genus. To explore that possibility, we have to follow the trail out of Africa and into the broader stage where these early experiments in being human first began to collide. When that trail leaves Africa and crosses into Eurasia, one region in particular turns into a maze. Asia, on a map, it looks like a single landmass, a broad expanse stretching from the Middle East to Japan, from Siberia down to the islands of Indonesia. But in evolutionary terms, Asia isn't one place. It's hundreds of ecological worlds stitched together. Deserts, monsoon forests, high plateaus, temperate plains, tropical coastlines. Mountain chains that act like walls, river valleys that function as highways, ice sheets that appear and vanish with the shifting climate. That kind of landscape does something very specific to populations. It breaks them up, reconnects them, breaks them up again. Over hundreds of thousands of years as climates warmed and cooled, corridors opened and closed. The Siberian steppe was sometimes a frozen barrier, sometimes a rich grassland. The deserts of Central Asia swelled during dry phases, then shrank when monsoons shifted. Sea levels dropped and rose, turning island chains into land bridges and then back into scattered specks of rock. Every time conditions changed, hominin groups moved. They followed herds into newly green regions. They retreated to refuges when deserts expanded. They crossed exposed continental shelves when the oceans fell, and every time they moved, they had a chance to encounter whoever had gotten there first. That's why many researchers think Asia, more than any other continent, hosted overlapping human and human-like populations for extraordinarily long stretches of time. The genetic clues from Denisovans are the clearest example of this patchwork. We only have a few physical remains we can confidently call Denisovan: a finger bone, a few teeth, a fragmentary skull piece from Denisova cave in Siberia, and a jawbone from the high plateau of Tibet that, based on proteins, looks Denisovan-like. That's it. A tiny collection you could fit in a small box. And yet, when you look at modern DNA, Denisovan ancestry pops up like scattered sparks across an enormous area. People in East Asia, in mainland Southeast Asia, in the Philippines, in Papua New Guinea, in parts of Australia, in some South Asian populations, many carry small but detectable fragments of Denisovan-related DNA. Not always the same kind either. Some groups like Papuans and indigenous Australians carry a very strong Denisovan signal that looks different from the weaker, more diffuse signal in East Asia. That pattern suggests there wasn't just one Denisovan group sitting in a single cave. There were probably multiple Denisovan populations, diverging from each other, adapting to local environments, moving in and out of contact with modern humans and with each other. A mosaic of related but distinct eastern hominins scattered from Siberia to the islands beyond Wallace's line. But if Denisovans were widespread, where are all their bones? That's where the story gets complicated. Asia's fossil record is uneven. Some regions, like parts of China and Indonesia, have produced numerous hominin fossils. Others, like much of mainland Southeast Asia, have barely been sampled, or sites have been destroyed by erosion, development, or simply never looked at with modern methods. And the fossils we do have from Asia rarely come with DNA. Warm, humid climates destroy genetic material quickly. Many key sites sit in exactly the wrong conditions for long-term DNA preservation. So instead of sequences, we're left with shapes. Skull fragments, jawbones, teeth, partial limb bones. When paleoanthropologists look at these, they often see something frustrating. Mosaics, faces that look partly like archaic Homo erectus, and partly like more recent humans. Teeth that share features with Neanderthals, but not all of them. Skulls that are overall modern-looking, but with a few very primitive traits that seem out of place. Think of sites like Dali and Jinshan in China, or the enigmatic fossils from Shujiao, or the remains from the Philippines and Flores that show odd combinations of features. Some of these get provisional names, some are left in broad categories like archaic Homo, others are debated endlessly in the literature. Are they early forms of Homo sapiens moving east? Are they late surviving descendants of earlier migrations like Homo erectus? Are some of them Denisovans? Or are we looking at mixtures? Populations that already carry genes from multiple branches, blending traits in ways our tidy species labels can't capture? Right now, we mostly don't know. The obvious temptation is to say, this weird fossil in China or Southeast Asia must be Denisovan. It's archaic. It's in the right region. Denisovan DNA shows up in local modern populations it seems to fit. But without genetic material from the fossil itself, that's just a guess. Morphology alone, the shape and size of bones, doesn't give a unique fingerprint for Denisovans yet. The few Denisovan remains we do have are mostly fragmentary and from limited anatomical regions. We don't know what a full Denisovan skull looked like. We don't know their typical body proportions. We don't even have a complete jaw with teeth and face attached. Trying to assign every odd Asian fossil to Denisovans would be like trying to match random, unlabeled keys to a lock you haven't actually seen. It might work by chance, but you wouldn't want to bet a grand theory on it. Instead, researchers treat these fossils as part of a broader pattern. Asia as a long-term meeting ground where multiple hominin lineages overlapped, diverged, and sometimes merged, Climate cycles and geography help us imagine how those meetings might have unfolded. During glacial periods when ice sheets grew and sea levels fell, land bridges emerged. The Sunda Shelf connected much of Southeast Asia to the Asian mainland. The Sahul Shelf joined New Guinea and Australia into a single landmass. Cold, dry conditions turned some regions into harsh steppe, but opened up new routes elsewhere. In warmer, wetter interglacials, forests spread. Deserts shrank. Northern corridors became more passable. Populations that had been isolated in refuges, coastal pockets, river valleys, mountain foothills, could expand outward, bumping into others doing the same. Over and over, Asia would have produced contact zones. The Levant, where modern humans and Neanderthals met. The Altai region, where Neanderthals, Denisovans, and later modern humans all left traces in the same cave. The mountainous belts of Central and East Asia, where high-altitude Denisovan-related variants show up. The island chains of Southeast Asia, where different human groups may have arrived in separate waves, each finding some earlier inhabitants already there. In each of these zones, the possibilities are similar. Sometimes, one group might push another out, directly or indirectly. Sometimes they might occupy slightly different niches, forest versus open plain, lowland versus highland, and coexist with limited contact. Sometimes they might interbreed, leaving a genetic legacy much larger than their demographic footprint. That's where forgotten lineages can hide. If a small regional group contributes just a few % of its genes to a larger incoming population, and then disappears as a distinct culture and morphology, its bones might be rare, its tools indistinguishable from its neighbours, its name never recorded. In areas with poor preservation and limited excavation, the odds of finding clear skeletal evidence drop even further. Yet the DNA can persist. As with Denisovans, modern genomes in Asia and Oceania are full of signals that don't map cleanly onto the fossils we know. Deep splits, unusual haplotypes, introgressed segments that look archaic but don't closely match Neanderthals or the sequenced Denisovan from Siberia. Some of those signals may come from unsampled Denisovan branches. Others may represent local archaic populations that never got a catchy label, groups that split off early, hung on in pockets of Asia, and then were absorbed without leaving a neat skeleton in a museum drawer. On the surface, Asia's human past looks like a scattering of puzzles pieces that don't quite fit, but if you step back, you can see the outline of something larger, a continent that acted not as a single stage, but as a vast shifting network of rooms and corridors. In each room, different versions of human met, mixed, or passed by each other in the dark. To understand our forgotten lineage, we have to see it against that background, not as a lone ghost, but as one thread in a densely woven fabric of encounters stretching across the largest continent on Earth. And if that's what was happening across Asia and its offshore islands, then one region in particular becomes crucial, a place where the Denisovan signal is strongest, and where the genetic echo of those encounters still rings the loudest today. When you follow that echo to its loudest point, you end up far from Siberian caves and Tibetan plateaus. You end up in Oceania. Genetically, one of the strangest facts about Denisovans is that their clearest legacy isn't in Siberia or northern China. It's in people thousands of kilometers away, in New Guinea, in parts of island Southeast Asia, in indigenous Australians, and some nearby island groups. Papuan populations, in particular, carry some of the highest levels of Denisovan-related ancestry known, on the order of a few % of their genome. That doesn't sound like much, but in population genetics, it's enormous. It's several times higher than what you see for Denisovan ancestry in East Asians, and it isn't just more of the same either. Those Denisovan-like segments in Papuans look somewhat different from the Denisovan-like segments in, say, Han Chinese or Japanese populations. When you compare them, they don't all trace back neatly to the same branch of the Denisovan family tree. That pattern suggests something important. Instead of a single clean episode where modern humans met a uniform Denisovan population and then carried that DNA out into the world, we're probably looking at multiple episodes involving Denisovan-related groups that were already differentiated from one another. So how do you get a strong, distinct Denisovan signal all the way out in places like Papua and northern Australia? There are a few broad scenarios researchers discuss. One is that the key encounters happened on the Asian mainland, before the ancestors of today's Papuans and indigenous Australians ever stepped onto the islands. In this view, a group of modern humans leaves Africa, moves through South or Southeast Asia, interbreeds with Denisovan-like populations along the way, and then a subset of that already admixed group pushes onward toward the far eastern edge of the Old World. Another scenario shifts the meeting point further along the route. In this version, some Denisovan-related populations had already spread into island Southeast Asia, maybe surviving in refuges on what was then the Sunda Shelf, or on isolated islands as sea levels rose and fell. Modern humans arrive later, encounter these eastern Denisovans closer to present-day Indonesia or the Philippines, and pick up genetic material there before continuing into Sahul, the combined landmass of ancient Australia and New Guinea. Both possibilities are compatible with the overall data. Neither is nailed down. What's clear is that by the time the ancestors of present-day Papuans and indigenous Australians reached Sahul, they were not pure descendants of a single African expansion. They already carried Denisovan-like DNA, and once they crossed that final water barrier into their new world, something else happened that matters a lot for how clearly we can see that signal today. They became isolated. This is where island biogeography steps in. When a small group of people reaches a relatively isolated landmass and then gets cut off from large-scale gene flow, their genetic makeup starts to drift. Some lineages increase in frequency by chance. Some segments of DNA stay around simply because there's nobody new arriving to dilute them. If those pioneers already carried a few percent Denisovan ancestry when they entered Sahul, that ancestry had nowhere to go but into their descendants. Generation after generation, it could be shuffled, trimmed by selection, or boosted in particular regions of the genome, but it wasn't being swamped out by continuous migration from other continents. Over tens of thousands of years, that isolation acts like an amplifier. The Denisovan signal that might be faint in a large, well-connected population can remain sharp and distinctive in an island population with limited outside input. Genetic drift, founder effects, and the demographic history of Oceania all combine to preserve and highlight those archaic segments. At the same time, selection could be quietly working on some of them. We already know that certain Denisovan-derived variants are associated with specific adaptations in other parts of Asia, most famously a gene variant influencing how the body handles low oxygen at high altitude in Tibetans. That variant looks Denisovan-like, and it's strongly favoured in people living on the Tibetan plateau where oxygen is scarce. Something similar might have happened with immunity, metabolism, or other traits in Oceania. If some Denisovan-related alleles happen to confer an advantage in local conditions, say, in dealing with particular pathogens in tropical environments, they could have been favoured by natural selection, becoming more common than you'd expect by chance alone. But here we have to be careful. It's tempting to point to every unusual genetic variant in Papuans or other Oceanian groups and say, aha, Denisovan superpowers. The reality is messier. Many adaptive candidates are still under investigation. Some that look archaic may turn out to be variants that simply evolved within modern humans. Others may trace back to Denisovan-like ancestors we haven't sampled yet, rather than the specific Denisovan genome sequenced from Denisova cave. In other words, even within the category Denisovan DNA, there may be multiple sources. That leads to another complication. The idea of a single Denisovan event may itself be misleading. Genetic modelling often starts with simple assumptions. One pulse of admixture here, one there, because that makes the math tractable. But as data accumulate, those simple models tend to fracture. Signals in Oceania, in East Asia, in South Asia, in the Philippines and beyond don't line up perfectly as products of one encounter with one homogeneous Denisovan group. Instead, they hint at a family of related archaic populations spread across Eastern Asia and its islands, mixing with modern humans at different times, in different places under different circumstances. The high Denisovan-related ancestry in Papua and neighbouring regions isn't just a curiosity, it's a clue that some of those archaic populations were present deep into the island world, not just on the northern steppes or Tibetan plateaus. It suggests a Denisovan-like geography that reaches from Siberia through Central and East Asia, down into Southeast Asia and quite possibly onto islands that were stepping stones towards Sahul. And if Denisovan-related groups could make it that far, or if their genes could, then the forgotten lineages implied by other odd genetic signals might have had similarly wide, patchy ranges, small populations in hidden corners, leaving durable traces in the genomes of people who managed to reach the end of the habitable world and stay there. Oceania, in that sense, is not a distant outpost at the edge of the map. It's one of the sharpest mirrors we have for the deep, tangled relationships between modern humans and the archaic cousins they met along the way, and for the possibility that some of those cousins were never fully captured by the names we've given them. To see how that plays out, it helps to look back at a lineage we think we understand much better, one that left actual skeletons and tools scattered across Europe, and yet still manages to surprise us every time we look more closely. Neanderthals, on paper, they're the opposite of a ghost lineage. We have skulls, skeletons, caves packed with tools, traces of fire and food and pigments. We've sequenced multiple Neanderthal genomes from different times and places. If any archaic human should feel known, it's them. And yet, when you look at Neanderthals through the lens of genetics, they turn out to be one of our best reminders of how much you can miss, even when you think you're looking straight at the answer. The first big surprise was simple and seismic. Neanderthals didn't just go extinct and leave us their bones. They left us their DNA. When researchers compared the first high-quality Neanderthal genome to modern humans, they saw something unexpected. People today whose ancestry traces mostly to Europe, Asia or the Americas carry small but consistent chunks of Neanderthal-like DNA scattered through their genomes. People whose recent ancestry is purely sub-Saharan African, in contrast, have far less. That pattern is how we know there was admixture, interbreeding between Neanderthals and the ancestors of non-African populations after modern humans left Africa. Those archaic pieces don't match any known African variation. They line up with Neanderthals. This basic before and after contrast, Africa versus the rest of the world, is the same kind of logic scientists now apply when they go hunting for other more mysterious lineages. You don't have to know what the ancestor looked like. You just have to see that some living populations carry segments of DNA that don't fit the usual tree. Neanderthals gave us the template, but as more genomes were sequenced from different modern populations, the pattern stopped being neat and uniform. Not everyone outside Africa has the same Neanderthal dose, and not everyone has the same Neanderthal segments. East Asians, for example, tend to have slightly more Neanderthal ancestry on average than Europeans, even though Neanderthal fossils are famously dense in Europe. Some populations in the Near East have distinct archaic segments that look like they came from early encounters. Others show signs of later pulses. When you zoom in, the mosaic becomes even more intricate. Different regions of the modern human genome show different histories. Some chunks clearly trace back to Neanderthals that were closer to the individuals sequenced from European sites. Others look a bit more like the Neanderthals sampled in Siberia. Still others suggest contributions from Neanderthal populations we haven't directly sequenced yet. That regional variation implies something important. There wasn't one single night in one single valley where modern humans met Neanderthals, and then the story was over. It looks more like multiple episodes in multiple places involving Neanderthal groups that were themselves regionally distinct. Somewhere in the Middle East or Western Asia, early dispersing modern humans may have picked up one set of Neanderthal segments. Later waves moving through Central or Eastern Eurasia may have encountered different Neanderthal populations, leaving a slightly different genetic fingerprint. This is why geneticists talk about pulses of admixture in the plural. Those pulses leave behind arcs of DNA that can be dated roughly by how chopped up they are. Long, continuous Neanderthal segments suggest more recent mixing. Short, fragmentary ones suggest older events, where recombination has had more generations to break them apart. That same logic is now applied to the unknowns. When researchers see archaic-looking fragments in African genomes, or unusual patterns in parts of Asia that don't match Neanderthals or Denisovans, they use the Neanderthal case as a kind of calibration. How long are the segments? How are they distributed across populations? Do they point to a single encounter, or repeated contact, or ongoing low-level gene flow over thousands of years? Neanderthals also taught us that not all archaic DNA is treated equally by evolution. If you look across the modern human genome, Neanderthal ancestry isn't sprinkled randomly. Certain regions are relatively rich in Neanderthal-derived fragments. Others are oddly empty. There are deserts where Neanderthal DNA is almost entirely missing. That tells us selection, natural selection, has been at work. In some locations, Neanderthal variants appear to have been harmful in the context of modern human biology. Genes involved in brain development, testes function, or certain aspects of early embryonic development, for instance, often show strikingly low levels of surviving Neanderthal input. The simplest interpretation is that archaic variants in those regions tended to reduce fertility or fitness when introduced into modern human genomes, so over time they were purged. Elsewhere, the story is the opposite. Certain Neanderthal-derived alleles seem to have offered advantages, at least in particular environments. Variants related to skin physiology, hair, and immune responses pop up again and again as candidates. These may have helped incoming modern humans cope with Eurasian pathogens, climates, and light levels. In some cases, the Neanderthal versions rose in frequency because they were better suited to local conditions than the versions carried out of Africa. This pattern of retention and removal, of some archaic segments being favoured, others being filtered out, is exactly what you'd expect when two long-separated populations merge and then adapt together to new environments. It's also the kind of pattern you look for when you suspect there was admixture with a lineage you can't see directly. If a ghost ancestor contributed to modern humans, its DNA wouldn't all survive intact. It would be broken up, shuffled, and then selectively trimmed or amplified over thousands of generations. You'd expect deserts and peaks, just like with Neanderthal ancestry, only harder to interpret because you don't have an actual reference genome to match against. And this is where Neanderthals become a mirror for our ignorance. Even with multiple Neanderthal genomes in hand, even with hundreds of modern populations sequenced, there are still arguments about the details. How many pulses of Neanderthal admixture were there? 2, 3, more. Were some of them limited to small regions? Did some Neanderthal populations contribute to one branch of modern humans but not another? How much of what we call Neanderthal DNA is actually from unsampled sister groups that lived alongside them? Our best sampled archaic lineage still leaves blurry edges. We can say there was mixing. We can estimate rough dates. We can identify regions where selection seems to have mattered. But we still debate the exact roots, the demographic histories, the extent to which different Neanderthal groups were involved. If that's what the picture looks like when we have bones, tools, and high-coverage genomes, you can imagine how much fuzzier it becomes when the lineage in question left almost nothing above ground. Neanderthals, in that sense, are practice. They show us how admixture looks when it's real. They show us how tangled the signal becomes when there are multiple encounters, structured populations, and hundreds of millennia of subsequent evolution. They show us that even known lineages can hide unsampled diversity, and that what we call Neanderthal DNA may in part be a stand-in for nearby cousins we haven't met yet. So when geneticists talk about ghost lineages, about ancestral populations that show up only as statistical shadows, they're not speculating in a vacuum. They're taking the tools sharpened on Neanderthals and turning them towards signals that don't line up with any named group. And if the Neanderthal story is any guide, those ghosts are unlikely to be simple, singular entities. They may be families of related populations, scattered across regions, meeting and mixing with modern humans more than once, leaving behind patterns that are only now starting to come into focus. To see how confusing that can get on the ground, you have to step away from DNA for a moment and look at the artifacts themselves, the tools, ornaments and traces of behaviour that litter caves and riverbanks, waiting to be matched to the right makers. When archaeologists first started pulling stone tools out of the ground, it was tempting to think each distinctive style mapped neatly onto a particular kind of human. You see a certain kind of hand axe, you say, that's Homo erectus. You see a particular blade technology, you say, that's modern humans. You find a chunky flake-based toolkit in Europe, you say, classic Neanderthals. It's a comforting picture. Every artifact has a maker. Every maker sits in a labelled box, but stone doesn't cooperate with our love of labels. A stone tool is, at its core, a solution to a problem. How do you cut hide, butcher meat, carve wood, pierce a thick skin, crack a bone? Many of those problems are universal, and the physics of sharp edges don't change from species to species. Give two different human groups the same kind of rock, the same kind of prey, the same constraints of cold or heat, and there's a good chance they'll converge on very similar shapes. A pointed flake is an efficient perforator, whether you're a Neanderthal, a Denisovan, or an early Homo sapiens with big ideas. This is what makes it so hard to look at a layer of tools and say, with confidence, who made them. Even the famous European Mousterian toolkit, long treated as the quintessential Neanderthal signature, turns out not to be exclusive. In some regions and time periods, anatomically modern humans seem to have used very similar methods. In other places, transitional industries blur slowly from Neanderthal style to modern style with no clean break. The same goes for older technologies. Simple flakes and cores take a rock, smash it, use the sharp bits, were made by multiple hominin species over well 1 million years. Hand axes appear in Africa, Europe and Asia, associated at different times with different members of our family tree. So when geneticists tell us there was an unknown lineage in a region, and archaeologists go back to the same caves and river terraces, there usually isn't a neat drawer full of ghost lineage tools waiting to be opened. There's just a long, overlapping sequence of similar technologies, tweaked, retweaked, reinvented. That's convergent innovation. Different groups, sometimes separated by hundreds of thousands of years, independently stumbling onto comparable solutions. But every so often the artifacts hint at something more than coincidence. You might find a distinctive tool type appear suddenly in a region where it hadn't existed before, without the gradual build-up you'd expect from local experimentation. Or you might see exotic raw materials, obsidian from a distant volcano, marine shells from a faraway coast, appearing in inland sites that show no direct access to those sources. Those patterns suggest contact, not just parallel thinking, but learning, borrowing, trading. In parts of Eurasia, for example, some archaeologists see technologies that look like hybrids, elements of typically Neanderthal preparation, mixed with blade and ornament traditions more often associated with early modern humans. There's debate about how to read this. Are these modern humans copying Neanderthal tricks? Neanderthals imitating newcomers? Independent groups co-evolving under similar pressures? Now add the possibility of a third unsampled lineage into that mix, and the waters get even murkier. If a ghost population shared landscapes with Neanderthals and early modern humans, their culture might have been absorbed, just like some of their genes. Tool shapes and techniques can be copied without leaving any trace of who first thought of them. Once an idea is loose in a network of groups, it doesn't carry a label saying, invented by Lineage X, 250 kilometers to the east. The same ambiguity haunts discussions of symbolic behavior. When you hear about beads, pigments, engravings, or possible art in archaic contexts, it's natural to think, aha, this must mark modern humans, or this must be the moment we became us. But the evidence refuses to line up neatly with that kind of story. Neanderthal sites have yielded perforated shells with pigment residues, possible pendants, and structures built from stalagmites deep inside caves. Some Middle Stone Age African sites associated with early Homo sapiens show careful use of red ochre, cross-hatched engravings, and potential mixing of pigments and binders that looks like paint preparation. Then there are more ambiguous finds, objects collected and cached for no obvious practical reason, bird bones with cut marks that might be related to feather extraction for decoration, accumulations of coloured minerals in contexts we don't fully understand. Are these definite symbols? Or are we projecting symbolism onto behaviours that might have had more mundane explanations, practical, aesthetic, ritual, in ways we can't pass? Reasonable scholars disagree, sometimes fiercely, because the line between functional and symbolic isn't always clear even in living cultures, let alone in sites tens or hundreds of thousands of years old. This is where archaeology and genetics can either hold hands or pull in opposite directions. DNA can tell us that two groups met and mixed. Archaeology can show that right around the same time, tool traditions in that region shift, or raw materials begin to travel farther, or pigments and ornaments suddenly appear in layers where they weren't present before. When those signals line up, you get a powerful narrative. New people arrive, new genes enter the population, new ideas spread through the toolkit. But sometimes the stories don't match. Genetic evidence might suggest prolonged contact in a region, yet the material culture record looks stubbornly continuous, with no obvious intrusion or hybridization of styles. Or the reverse can happen, a dramatic cultural turnover in the artifacts, with no clear genetic break in the skeletons we've sampled so far. In those cases, either the archaeology is only seeing part of the picture because organic technologies like baskets, clothing, or wooden tools have vanished, or the genetics is undersampled, missing lineages that never got preserved well enough for us to sequence. And hovering behind all of this is the idea of forgotten, not just as a biological fact, but as a cultural one. A lineage can disappear genetically, its DNA diluted and scrambled until only statistical traces remain. Culturally, something similar can happen, but even faster. The songs, stories, and rituals of a small population can be borrowed by neighbours, modified, translated into new contexts, and eventually remembered under someone else's name or with no name at all. Rituals of the dead, ways of reading the sky, patterns of movement across a harsh landscape. Any of these could have originated in groups that left few descendants, yet lived on in the knowledge and habits of those who followed. When we talk about a forgotten human lineage, then we're probably understating the case. We may have forgotten their bones, their faces, their languages, but parts of how they lived, how they hunted, healed, crafted, and perhaps how they imagined the world, may still be echoing through traditions we casually assign to early modern humans or Neanderthals, simply because those are the names we know. The tools in the ground don't correct us. They just sit there, anonymous, sharp edges, waiting. To understand how a lineage can vanish in body yet persist in fragments, both in our DNA and in our cultures, you have to think about what it means to go extinct when the boundaries between groups are porous, and when meeting doesn't always mean replacing. Sometimes it means being absorbed. When people hear that Neanderthals went extinct or that a mysterious ghost lineage died out, it sounds final. A clean break. One day they're here walking across the steppe or along a forest edge. The next they're gone, like a light switched off. But in a world where groups are small, mobile, and capable of interbreeding, extinction doesn't always mean what it sounds like. Imagine a valley where two groups live side by side for a few thousand years. One group is big, maybe a few thousand individuals spread across many camps and seasonal sites. They have wide trade networks, marriage ties that stretch over long distances, and technologies that help them cope with bad years. The other group is small, a few hundred people at most, clustered in a handful of refuges. If the climate shifts, prey herds move, or disease sweeps through the region, which group is more likely to weather the storm? Even if the smaller group is skilled and resilient, they're more vulnerable to bad luck. A couple of harsh winters in a row, a new pathogen introduced through contact, a series of poor hunting seasons, and their numbers can fall below a threshold that's hard to recover from. But that doesn't necessarily mean they vanish without a trace. Members of that smaller group can marry into the larger one. Children can be raised in the camps of their mother's people or their father's people, learn the language and customs of whichever side has more food, more security, more allies. Over a few generations, the original small group may lose its distinct identity. No one calls themselves by that old name anymore. The clan markers, the particular way they painted their bodies, the stories that trace back to that lineage fade. Yet the DNA of those people hasn't gone anywhere. It's just been diluted into a much larger gene pool, like a drop of dye spreading through a lake. Geneticists sometimes call this demographic swamping. It's not conquest in the Hollywood sense, with one group marching in and slaughtering another. It's a long, slow process where sheer numbers, combined with mobility and mating patterns, absorb smaller populations into bigger ones. Climate instability can speed this up. In a stable environment, neighbouring groups might coexist for long stretches, each holding on to their territories, maintaining distinctive traditions. But when rainfall patterns flicker between extremes, when coastlines flood and retreat, when forests thin into grassland and then close up again, groups are pushed into motion. Refuges shrink and expand. Migration routes cross in new ways. Populations that once rarely met are suddenly sharing the same watering holes, the same narrow passes, the same sheltering caves. Under those conditions, ecological competition becomes more direct. 2 or 3 different hominin lineages might all be targeting the same herds, the same nut groves, the same river crossings. The one with better information networks, more flexible hunting strategies, or simply more people in the right place at the right time gains an edge. Disease complicates this even further. When previously separated groups meet, they bring their pathogens with them. A virus that's mild in one population because they've lived with it for generations, can be devastating when it jumps into another that has no prior exposure. The archaeological record doesn't let us see individual epidemics clearly, but the biology of disease makes it hard to imagine deep human history without these invisible shocks. A small lineage caught between environmental change, competition, and waves of unfamiliar diseases might see its numbers crash. But again, that doesn't always play out as a sharp, clean ending. Survivors may join neighbouring bands simply because that's where food, mates, and protection now lie. Mobility and social networks decide which lineages are more likely to supply those havens. Groups with long-distance ties, kin in another valley, trading partners over the mountains, friendly faces at a faraway lake, have more options when local conditions deteriorate. They can move, split, and rejoin, seed new camps in less crowded regions. Ghost lineages, by definition, are ones we haven't sampled directly in the fossil record. We mostly know them as statistical shadows in our genomes. That alone suggests something about their fate. If a lineage truly left no descendants at all, its DNA would stop at the last individual who carried it. There would be no trace for us to detect in living populations. The very fact that we can see ancient, divergent DNA threads woven into the genomes of people today tells us that absorption, not pure extinction, has been common. So a group can vanish as a named, discrete entity while still persisting, in fragments, inside the bodies of those who came after. There's a subtle but important distinction here: disappearance from genes versus disappearance from culture. From a genetic point of view, a single child is enough to carry a lineage forward. A handful of births, scattered over a few generations, can preserve rare variants for hundreds of thousands of years if they happen to land in populations that grow and spread. Culturally, survival is more demanding. To keep a language alive, you don't just need a couple of grandchildren. You need a continuous community of speakers, passing the sounds and structures on day after day. To maintain rituals, ecological knowledge, or tool-making secrets, you need shared practice, shared memory, a group that sees itself as the proper custodian of those ways. When a small lineage is absorbed, its genes can flow almost invisibly into the expanding population. Some bits will be favoured by natural selection, better immunity, improved adaptation to altitude, resistance to cold or heat. Others will drift along neutrally, neither especially helpful nor harmful, just part of the background variation. Its culture, though, has to compete in a different arena. Some practices are easy to borrow without carrying their original identity. A hunting technique that works, a clever snare, a new way of hafting stone to wood. These can spread quickly through neighbouring groups. Over time, people may forget where the idea came from. It just becomes how we do things. Other cultural elements are more tightly bound to group identity, origin stories, sacred places, names for the spirits in a particular cave, rules about whom you can marry. These may be dropped or transformed when people shift their allegiance, marry into larger networks, or relocate to new landscapes. So a forgotten lineage might live on in our genes while its language, myths, and social structures leave little trace, or survive only in altered, unrecognizable forms within the traditions of those who absorbed them. This is why extinction stories in human evolution are often too tidy. It's tempting to say Neanderthals died out around this date. Denisovans disappeared from this region by that time. A ghost population split here and went extinct there. The graphs on a paper need lines that start and lines that end. Reality, as far as we can tell, was messier. Ranges contracted and expanded. Groups fragmented and reformed. Some lineages dwindled in one area while flourishing, under another name somewhere else. Contact wasn't a single encounter but a mosaic of meetings, some hostile, some friendly, some intimate. When geneticists draw a branch of the human tree and then mark it with a little cross to show extinction, what they often mean is that we can no longer trace a continuous separate line from that branch to a living population. It doesn't necessarily mean there were no descendants, only that those descendants were folded back into other branches. And when we imagine the last member of a lineage sitting alone on a cold hillside, looking out over a world that no longer has a place for them, we might be picturing the wrong kind of ending. It may be that the real finales were quieter, a series of marriages, adoptions, guest seasons spent with allies that became permanent moves. Children who grew up hearing two sets of stories then passed on only one. Genes sliding into new social worlds, while the old names slipped out of memory. For the mysterious lineages we're chasing through ancient DNA, this difference between extinction and absorption matters. It changes the kind of questions we ask, not just when did they disappear, but where did they go inside us? How much of them still lingers, hidden in our biology and in the deep, untraceable roots of our cultures? To get closer to those answers, researchers don't just look at who mixed with whom, but when. The timing of admixture events, those pulses of genetic blending, can tell us whether a lineage was holding its ground, retreating into refuges, or already in the process of being swallowed by larger populations. And that takes us to one of the trickier tasks in this whole story. Figuring out when, in that long twilight between distinct identity and full absorption, these meetings actually happened. When geneticists say something like, modern humans mixed with this archaic lineage around 50,000 years ago, they're not reading a date off a fossil label. They're building that estimate from patterns in DNA, and those patterns are slippery. One of the main tools they use is something called a recombination clock. Every time a child is conceived, they don't just get a solid block of chromosomes from each parent. Those chromosomes have been shuffled. Segments of DNA are swapped and recombined, chopping long stretches into smaller pieces, generation after generation. If your ancestor had a Neanderthal great-great-great something or other, they passed down chunks of Neanderthal DNA. At first, right after the interbreeding, those chunks would have been long, big, uninterrupted stretches, easily recognizable as from that other population. But each new generation of recombination cuts them up a little more. So if you look at the genomes of living people, you can measure how long those archaic segments are. Longer on average means fewer generations since the admixture. Shorter on average means more generations, more chopping, more time. It's like looking at a mosaic and trying to guess how many times it's been broken and reglued. But a recombination clock is only a clock if you know how fast it's ticking. That's where things get complicated. To turn this many generations ago into this many years ago, you have to assume something about how long a generation is. Do you use 25 years? 29? 32? Small changes make a big difference when you multiply them by thousands. If a model assumes a 25-year generation, an event might be dated to roughly 50,000 years ago. If another researcher prefers 30 years per generation, the same number of generations suddenly corresponds to 60,000 years. Nothing changed in the DNA, only the conversion factor shifted. The same thing happens with mutation rates. Another way to estimate time is to count new mutations, tiny copying errors that appear each generation. If you know roughly how many mutations tend to accumulate per generation, you can estimate how many generations separate two lineages that share a common ancestor. But mutation rates aren't perfectly fixed. They can differ slightly from one population to another, and they can be measured in different ways. Some estimates come from comparing humans and chimpanzees and dividing by fossil-based divergence times. Others come from sequencing large numbers of modern families and directly counting new mutations between parents and children. Those methods don't always agree, and the differences ripple through everything. Use a fast mutation rate, and the split between two lineages might be placed at 600,000 years ago. Use a slow rate, and it shifts to 800,000 or 900,000. Again, the genomes are the same. The timeline stretches or compresses depending on which ruler you use. Ancient genomes help to anchor these drifting clocks. When researchers can sequence DNA from a bone that's been independently dated, for example, by radiocarbon or other geological methods, they suddenly have a fixed point. They know that this individual lived, say, 45,000 years ago, and they can see exactly how many mutations separate that genome from modern ones. Those anchors allow them to recalibrate their mutation rates and refine their recombination models. Sometimes this pushes admixture dates earlier than we thought. Sometimes it pulls them forward in time, closer to the present. It's one reason why you might see headlines a decade apart that seem to contradict each other. Early studies might say, modern humans left Africa 60,000 years ago. Later analyses with more ancient genomes and better calibration might suggest 70,000 or 80,000, or even multiple stages of dispersal. On top of all this, the models themselves matter. Different statistical methods make different assumptions about how populations behaved in the past. One model might assume a single sharp pulse of admixture. Two groups meet once, exchange genes for a short time, then separate. Another might allow for trickles and drips of interbreeding over thousands of years, with populations splitting, rejoining, and exchanging migrants more than once. Feed the same genetic data into these different frameworks, and you can get different answers. Not because one is lying, but because each is asking a slightly different question about the past. Some methods are especially sensitive to recent events. Others blur recent details but capture deep, ancient structure. Some treat populations as cleanly separated until they mix. Others allow ghost lineages and unsampled branches to influence the results. All of that means we need to be careful about precision. When a paper reports that two lineages had mixed 47,300 years ago, it's tempting to imagine a specific generation, a particular winter or spring in deep time. But what the science really supports is something more like somewhere in this broad window, maybe between 40,000 and 55,000 years ago, with the peak probability near the middle, based on the assumptions we've made. The dates shift because the assumptions shift, and because new data, especially ancient genomes, force the clocks to be reset. So when you hear that a ghost lineage mixed with the ancestors of modern Africans around 200,000 to 300,000 years ago, or that Denisovan-related DNA entered certain populations around 45,000 to 50,000 years ago, it's better to think in ranges and scenarios, not single numbers. The point isn't to pin an exact year on a meeting that happened before our own species had a name. The point is to place these encounters in their proper chapters of the human story, before or after certain climate swings, earlier or later than key migrations, overlapping with one lineage's rise and another's decline. And once those time windows are sketched in, we can start to ask the deeper questions. Who was where, what the world looked like when they met, and how those brief moments of contact left signatures strong enough to survive in our DNA across hundreds of thousands of years. So far, our picture of this forgotten lineage has come mostly from bones, teeth, and the genomes we can squeeze out of them. But bones are rare, and intact DNA is rarer still. Most of the humans who ever lived left nothing behind that we can hold in our hands. That's why one of the most exciting frontiers doesn't start with skeletons. It starts with dirt. When archaeologists dig in caves and rock shelters now, they don't just collect artifacts and bones. They also scoop up the sediment itself, the compacted dust, ash, and microscopic debris that has settled layer after layer over tens of thousands of years. Hidden in that dirt are tiny fragments of DNA, shed from skin cells, hair, bodily fluids, and decomposing tissue. It's called sediment DNA or environmental DNA, and it lets scientists detect which species were present in a place, even when no bones survived. In some caves, researchers have pulled Neanderthal DNA and Denisovan DNA directly out of the floor sediments from layers where no recognizable hominin bones were found at all. Just from the genetic fragments they can say, a Neanderthal was here, or Denisovans used this cave, and match those traces to particular branches of the family tree. For a ghost lineage, that's huge. Imagine a site in Africa or Asia with stone tools that don't quite match known cultures, or hints of archaic anatomy from a single tooth. If sediment DNA from that layer shows a distinct hominin genome that isn't Neanderthal, isn't Denisovan, and isn't modern human, that would be a powerful clue that we're finally brushing up against the forgotten branch. But this method has serious pitfalls. Sediment is messy. DNA can move between layers when water percolates through, or when burrowing animals disturb the ground. Later occupants of a cave can shed far more DNA than earlier ones, drowning out faint ancient signals. And tiny modern contaminants, one careless breath, one drop of sweat, can masquerade as ancient humans if strict precautions aren't taken. On top of that, sediment DNA is usually broken into extremely short fragments, often too small to reconstruct full genomes. What we get are molecular whispers, enough to say some kind of hominin, sometimes enough to assign it to a broad group, but not always enough to map fine-grained relationships. When DNA fails completely, another frontier opens: proteins. Unlike DNA, which unravels relatively quickly over geological timescales, certain proteins can survive for hundreds of thousands, even millions of years in the right conditions. The field that studies these ancient proteins is called paleoproteomics. Bones and teeth are full of structural proteins like collagen. The exact sequence of amino acids in those proteins is shaped by an individual's genes, and by comparing those sequences across species, scientists can infer evolutionary relationships even when no DNA remains. This has already rewritten parts of the hominin tree. For some very old fossils where DNA is hopelessly gone, protein analysis has been able to say this jaw is closer to the Neanderthal Denisovan branch than to early modern humans, or this hominin sits outside all known groups. It's a lower resolution picture than a full genome, but it reaches much farther back in time. For a deeply divergent lineage that contributed only a trickle of genes to our ancestors, ancient proteins might be our only molecular evidence. A single tooth in North Africa, with a protein signature that doesn't match any known hominin, could be the first solid anchor for the ghost we only see in statistics. All of this is being turbocharged by better extraction and sequencing technologies. Early ancient DNA work needed frozen mammoths and permafrost. Now ultra-clean labs, improved chemical treatments, and more sensitive sequencing machines can pull usable fragments out of warmer, older, more degraded samples. Bones once written off as too old or too contaminated are being revisited and sometimes yielding surprises. Short read sequencing has been joined by newer methods that can handle longer fragments, or that are optimized to capture very particular stretches of interest, immune genes, mitochondrial DNA, or even specific archaic segments we suspect might be present. That expands the map of where we can look and what we can ask. On the computational side, the tools are evolving just as quickly. New demographic models can explicitly include ghost lineages, branches for which we have no direct genome, only their faint echoes in other people's DNA. Researchers can simulate thousands of complex scenarios, multiple waves of migration, repeated interbreeding, long-term isolation, followed by sudden contact, and see which patterns best match reality. Machine learning and AI methods are starting to help reconstruct ancient haplotypes, the original chunks of DNA that entered our lineage from Neanderthals, Denisovans, and possibly others. By scanning modern genomes for distinctive patterns of variation, algorithms can piece together what those archaic segments probably looked like when they first arrived. In some cases, these reconstructed sequences can be compared against partial genomes from actual fossils. When they line up, it validates the method. When they don't, when the inferred archaic sequence has no good match in any known ancient genome, that discrepancy itself becomes a clue. It hints that some of the DNA we carry comes from a population we still haven't sampled. Powerful as these methods are, they come with responsibilities. Ancient remains, cave deposits, and even sediments are not just scientific resources. They are part of cultural landscapes, sacred histories, and living traditions. Extracting DNA or proteins usually means destroying a tiny bit of what's left. A piece of bone, a grain of tooth enamel, a scoop of soil from a burial layer. Who gets to decide when that's acceptable? How do we balance the desire to solve scientific mysteries with the rights and beliefs of descendant communities? More and more, the answer is by asking first, by involving those communities from the beginning, and by sharing both the data and the story that emerges from it. Ethical guidelines now emphasize minimal destructive sampling, clear consent processes, and collaborative research designs where local voices shape the questions being asked. That matters here because the forgotten lineage we're hunting may be an ancestor not in the abstract, but of specific modern peoples. The moment we can finally put a genetic and archaeological face on this branch, it will become part of someone's identity, someone's origin story. So what would count as a decisive discovery? One possibility would be a well-dated fossil, perhaps in Africa or Western Asia, that yields either a partial genome or a distinctive protein profile which matches the ghost signal we've inferred from modern DNA. Its place in the tree would line up with the strange, deep splits we see in some African genomes or the odd archaic segments in Asia that aren't Neanderthal or Denisovan. Another would be a cluster of sediment DNA samples from multiple sites and layers, all pointing to the same unknown hominin population occupying particular regions during a specific time window. If those layers also show unique tool traditions or ecological niches, we could begin to link a genetic lineage to a way of life. Or the breakthrough might be mostly computational, a convergence of different models, sediment signals and protein data, all indicating that the same unsampled branch must have existed, with its own long history, its own migrations, its own encounters with our ancestors. When those lines of evidence finally cross and hold, the forgotten lineage will stop being a ghost in the equations and become a real, if still shadowy, people in our deep past. And then we'll have to ask a harder question, not just how we found them, but who exactly they were. When scientists talk about this hidden lineage, they're not conjuring fantasy ancestors out of thin air. They're trying to explain stubborn patterns in the data that won't go away, no matter how you tweak the models. If you strip it down to the strongest points, you get something like this. Some modern populations, especially in parts of Africa and Asia, carry stretches of DNA that look too old, too different, and too structured to have come only from the known branches, early Homo sapiens, Neanderthals, and Denisovans. These segments behave as if they came from a group that split off earlier, stayed separate for a long time, and then mixed back in. That's the signal. Everything else is an attempt to give it a face. One conservative possibility is that we're not looking at a single new species at all, but at extreme diversity inside groups we already know, especially the Denisovans. The Denisovan label comes from a single cave in Siberia. But the DNA from that cave already hints that Denisovan might be more like a whole cluster of related populations spread from Siberia down through East and Southeast Asia. Some of those branches split from each other almost as deeply as Neanderthals split from modern humans. In that view, the unknown lineage could be a very divergent Denisovan branch, one that lived in parts of Asia we haven't sampled yet. Their bones might be sitting in museum drawers, mislabeled as generic archaic Homo, their DNA too degraded for early methods to catch. To people living in island Southeast Asia or the highlands of New Guinea, they would have been real neighbours, trading partners, rivals. To us, they're statistical echoes in the genome. A second, more Africa-focused possibility is that the ghost lineage represents one or more archaic groups that stayed in Africa while Neanderthals and Denisovans moved out. Africa during the Middle Pleistocene was not an empty stage with a single cast. It was a whole continent of shifting environments, rivers opening and closing, forests expanding and retreating, savannas turning to desert and back again. Fossils from this time already show surprising variety, skulls and jaws that don't fit neatly into modern human or anything else. In that patchwork landscape, it's plausible that an early branch of the human family became semi-isolated in particular refuges, dense forests of Central Africa, the Ethiopian highlands, or coastal zones along vanished shorelines. They would have adapted to local conditions, developing their own body shapes, their own tool traditions, their own social networks. Every so often, climate shifts or migrations would bring them into contact with more typical Homo sapiens populations, leaving small genetic traces that still show up today. The most radical version is the Super-Archaic scenario. Here, the unknown lineage isn't just a side branch of our own near relatives, but a much older offshoot, maybe related to very early Homo populations that left Africa long before the ancestors of Neanderthals and modern humans. In this model, some of those early wanderers survived in remote pockets of Eurasia or Africa for hundreds of thousands of years, long after their cousins had vanished elsewhere. When our more recent ancestors expanded, they didn't just encounter Neanderthals and Denisovans. They also met these relic populations who had been evolving on their own for an extraordinarily long time. A few episodes of interbreeding would have been enough to tuck fragments of super-archaic DNA into our genomes, even if the populations themselves were small and eventually disappeared without leaving obvious fossils. Each of these possibilities comes with favoured habitats and lifestyles. A Denisovan-like branch might have thrived in rugged mountain zones and wooded uplands across Asia, specialising in high-altitude survival, forest hunting, and possibly even early seafaring along island chains. An African archaic group could have been masters of gallery forests and river corridors, or of humid tropical belts, where bone preservation is terrible and fossils rarely survive. They might have focused on fishing, plant collecting, or hunting smaller game ways of living that leave fewer dramatic fossil-rich kill sites, but still support stable communities. A super-archaic population, if it existed, may have been pushed into ecological margins, deep forests, isolated plateaus, or coastal refuges where later arrivals were slow to follow, places where a small, conservative way of life could persist for a long time with little change. Will we ever be able to point to one skull, one name, and say, that's them? Maybe not. Human evolution doesn't really work like a tidy staircase of species, each with a clear label. It's more like a braided river, channels splitting and rejoining, side branches drying up, new ones forming downstream. What genetics sees are relationships and splits, not neat boxes with capitalized names. A decisive discovery would be something like this: a well-dated genome, or at least a clear protein profile, from a fossil in a region and time window where the ghost signal is predicted. Its place in the tree would have to line up with the strange, deep divergences seen in living people's DNA. Its geography and age would have to match the admixture dates. And, ideally, we'd find similar evidence at more than one site. so it's not just a one-off outlier. Even then, we might still argue about what to call them. Are they a distinct species, a subspecies, a highly divergent population of something we already named? The labels may never be as satisfying as the story, because the deeper lesson of this forgotten lineage is not that there was one more box on the chart. It's that our past was never a simple ladder with primitive at the bottom and modern at the top. It was a web of connected populations, splitting, mixing and shifting across continents and climates. The ghost in our DNA is a reminder that we are not the lonely survivors of a clean-cut family tree. We are the latest moment in a long, tangled continuum of humans, all borrowing genes, ideas, and ways of living from one another as the world changed around them. So where does that leave our forgotten lineage? Not as a monster in the shadows or a missing rung on a ladder, but as one more thread in a very old tapestry. Ancient DNA has been quietly rewriting what the word human meant for most of our history. It used to mean us, and only us. A single species, modern and unique, rising cleanly out of a murky background. Now it means something wider. It means Neanderthals carving tools and burying their dead. Denisovan wanderers moving along islands and mountain chains, leaving survival genes in the lungs of Tibetan herders and the immune systems of Pacific islanders. It means African populations we don't yet have names or faces for, yet whose DNA still hums along inside millions of people. It means that being human was never about belonging to the right box or having the right skull shape. It was about belonging to a network of shared genes, shared skills, shared stories, told in languages we will never hear. There are a few things we can say with some confidence. We know that the people alive today are the product of multiple lineages mixing and remixed over hundreds of thousands of years. We know that at least one of those lineages has not yet been directly sampled in the fossil record, but still whispers through our genomes. We know that interbreeding wasn't a rare accident. It was part of how humans lived, moved, and adapted. And there is a lot that remains open. We don't yet know how many distinct populations contributed to the ghost signal, or what they looked like, or exactly where they thrived. We don't know whether they saw themselves as different, or just as another band of people over the next ridge. We don't know how they told their origin stories, or what they thought when strangers arrived speaking oddly, moving differently, carrying unfamiliar tools. Those answers may never come in full, but even the questions change how we see ourselves. Because if you follow the DNA trail all the way back, past nations and languages and labels, you reach a small, frightened set of survivors riding out ancient climate swings. You reach a world where the boundaries between us and them were porous, where every meeting between populations was a chance to exchange not only genes, but knowledge. When you lie down to sleep tonight, your body is carrying that history. Chromosomes fused in some long-ago crisis, variants tuned to old diseases, old diets, old skies, ghost fragments from people whose names are lost, but whose choices and chances helped make you possible. In that sense, there is no truly forgotten lineage. As long as any of their DNA remains, as long as any of their adaptations still help a child breathe at altitude, fight off an infection, or think just a little more flexibly, they're still part of the living human story. If you'd like to wander further into that deep past, I've put together a book exploring 12 of the most intriguing prehistoric secrets, from bottlenecks and ghost lineages to the first art and the first seafarers. You can read more about it and pick up a copy if you'd like through the website linked in the description. If you're listening as you drift off, feel free to let the details blur and just hold on to the shape of the story. A planet changing, small bands of humans adapting, separating, reconnecting, and slowly becoming us. Before you go, or when you wake up, I'd love it if you'd gently tap like, subscribe to the channel, and leave a comment telling us where in the world you're listening from right now. Each place, each voice, is another branch of that same ancient tree. And somewhere deep in the code of your cells, the forgotten lineage is there with you too, quiet, persistent, and still part of what it means to be human.